Phylum Ascetospora. What is Phylum Ascetospora? Phylum Ascetospora is a small phylum consist exclusively of parasitic protists. It is often characterized by spores lacking polar caps or polar filaments. In addition, Ascetospora are obligatory extracellular parasites. Acetosporians are primarily parasitic on the cells, tissues, and body cavities of mollusks. According to Wikipedia, Acetosporia is a group of eukaryotic parasites primarily infecting marine invertebrates. Within this group, haplosporids and paramecids, though morphologically dissimilar, consistently cluster together on molecular trees. Placed near the base of the cercozoa, both haplosporids and paramecids produce spores without complex structures, distinguishing them from similar groups. Let us differentiate haplosporid spores from paramecids. Haplosporid spores have specific characteristics, including a single nucleus and a unique opening covered by diaphragm or hinged lid. They go on to develop within the cells of their host, which are typically marine mollusks or annelids. The trophic cell stage within the host is multinucleated. Paramecids, on the other hand, are characterized by developing within the digestive system of marine invertebrates. Their life cycle involves internal budding, resulting in the production of multicellular spores. Next is the life cycle of Acetospora. So the life cycle of Acetospora may actually vary depending on its species. What I am presenting here is a generalized overview of its life cycle. So first is the infection and entry. Acetospora are obligate parasites, which means they rely on a host for survival. The life cycle often begins when the acetosporian spores come into contact with a suitable host or organism, usually a mollusk. Second is host penetration. The acetospores penetrate the host tissues, often targeting cells, tissues, and body cavities of mollusks. The third stage is the intracellular development. Once inside the host, the acetospores undergo development and the parasites often become intracellular, affecting the host cell. The next stage is the multiplication and formation of ineffective stages. The acetospore undergoes multiplication within the host, leading to the formation of ineffective stages. These stages are often responsible for spreading the infection to other individuals. Next is the release of ineffective stages. The ineffective stages are released from the infected host into the environment. This can occur through various means, such as the host bodily fluids or upon the death of the host. The sixth stage is the transmission to new host. The release ineffective stage aims to infect new hosts. This transmission can occur through direct contact between hosts or through the water in which the hosts live. And lastly, the completion of life cycle. The cycle continues as new hosts become infected, and the acetosporian parasites undergo reproduction and development within these hosts. Once again, I would like to take note that these stages may actually vary among different acetosporian species. For the last part of this lesson, I'm gonna provide some species of Acetospora. First, we have the Haplosporidium. Haplosporidium species are known to infect mollusks, causing disease such as MSX in oysters. These infections can be detrimental to aquaculture and wild mollusk population. The next species is the Marchiella. Marchiella species are protistan parasites that infect bivalve mollusks. They can cause disease such as marchiellosis, impacting oysters and mussel population. And finally, Bonamia austriae. It is a parasite that infects oysters, particularly Ostraea edulis, which is a species of flat oyster. Bonamia austriae causes a disease known as bonamiosis. The parasite has been historically considered to have circumscribed geographic range, primarily affecting oysters in the northern hemisphere. And that's all for our presentation. I hope you learned something. Thank you for listening and have a good day.